Okay, guys. Here we are. Okay, so I keep adding to the list. I won't go through the whole thing. But most recently, we've been talking about vesicular transport. Last time we talked about KDEL, the ear retention signal. We spoke about, so that's how ear resident proteins are brought back from the Golgi. We talked about the Golgi and that there's this processing of trimming and addition of sugars. And one of the things that's added in the Golgi is mannose-6-phosphate, so lysosomal proteins, and that sends them to the lysosome, not to be degraded, but because they're acid hydrolases that work in the lysosome. And then we were talking about the endocytic pathway, which is endocytosis, right? So we ended with this, and hopefully everyone did it. But we'll take a moment here. I'm going to try to use this thing. I never use this thing. I don't know how this is going to go. Uh-oh, who said good luck? Let's see.
It's harder than Jesus.
I'm going to see if I can do it. Otherwise, I'll use the old-fashioned chalkboard. So if you guys want, take a minute to bring your brains back to this pathway. Look at that. It's my hand. Okay. Okay, guys. Do you have your brains back? Okay, do you want to take a minute to regroup? Okay, so remember this was the draw out transport of lysosomal proteins from the Golgi to the late endosome. Make sure to include lysosomal protein with the correct modification, the correct receptor, vesicle formation, including adaptin and clathrin, and mechanism for protein binding in the Golgi and release in the late endosome. Okay? You guys are like, yeah. Okay. So I'm gonna go through it now because you guys, we had a few minutes at the end of the class the other day. Okay. So transport of lysosomal proteins from the Golgi to the late endosome. I guess that means, I hope. Oh, that's weird. Okay. Let's have let's put a Golgi here. Pancakes. I know there's more than that. And then let's call this the late endosome. Okay. Hey, it's getting confused, huh? I guess that's just how it's going to be. How do you guys feel about that? Okay. It's the best thing ever. Good. Okay. So first thing, we need a lysosomal protein that needs to go from where to where? Where's it going to start? In the Golgi. Technically, in the ER, right? ER. But we're going to pick it up over here. So here's your lysosomal protein, nicely folded. Looks like a mushroom. What do I need to add to it in the trans-Golgi network? Oh, that is so crazy. Guys, are you going to throw up? I just need to keep my hand out of there. Okay, mannose-6-phosphate. So, M6. Which lights? Turn off all the lights in the room. Someone might steal your wallet then. We need, they always have that security light on, just in case. Okay. Well, I think if I keep my hand here, we're okay. No? Okay. Well, let's just work.
There should be a NOS that you can like.
No, it's auto-focusing. That's the problem. Oh. it wants to autofocus. Let's just go with it. We're gonna go with it, and I'll periodically take my hand away. There's the mannose-6-phosphate. Okay, so what's gonna happen next? Okay, so how's it gonna get packaged into a vesicle? I hear people saying receptor. So what kind of receptor? The LDL receptor? Okay, mannose-6-phosphate receptor. Okay. So there's going to be that thing that's going to bind. Well, actually, technically, it's going to bind to the mannose-6-phosphate, but it's okay. So here's your mannose-6, that's a G, phosphate receptor. Okay. Oh my God, it's horrible, guys. See, chalk. It's all about chalk. Oh, I'm going to puke. Okay. Anyways. Okay, so then you need to be incorporated into a vesicle, which is, I've drawn this poorly, but let's see. So there's going to be a vesicle that's going to form. But how is the mannose-6-phosphate and receptor going to make it into that vesicle? What's it going to interact with? Adaptin, right? So we're going to actually have a whole bunch, because remember, it's not just one protein going in. And so we're going to have a whole bunch of little mushrooms, and there are M6Ps in there. And so that's going to bind to adaptin. Okay, and then what's that going to bind to? Clathrin. And so that's causing that curvature. And in reality, it's kind of happening, a little bit at a time and whatever. And that's causing that curvature. And so then what's it going to do? What? It's going to pinch off. Okay, so then you're going to have, then it's going to pinch off, and then you're going to lose your clathrin, but you're going to have your mannose-6-phosphate receptor bound to your lysosomal acid hydrolase, and then it's going to do what? Where's it going to go? To the endosome, right? So it's going to go there. And is it just going to, so how are these acid hydrolases going to get into the endosome? Are they just going to like pop out of the vesicle and slide through the membrane for the endosome? How's that going to get to there? Okay, so I'm hearing something about RAB, right? So you have RABs and snares, right? So you probably have a RAB protein, and that wasn't in the assignment, but that's okay. And you probably have snares, right? And so it's going to go, and then it's going to tether through RAB, which is in what bound state? GDP or GTP? GTP, because that's on. And so it's going to tether to the RAB effector, then the RAB's going to bind, and that's going to kind of bring it close somehow so that you've got the snares there. And they're going to kind of wind up together to help to make it fuse. Okay, and so what's in here? What are we doing? What's the point of this? What's in this receptor? Acid hydrolase that's tagged with what? Mannose-6-phosphate. And so then, notice it's kind of released in there, right? So now the question is, so now you have your endosome, so that's kind of this progression here. And so the pH, uh-oh. Nope. Whoop. So now you, oh dear. So now you have this endosome. What pH is the endosome in relation to the Golgi? Is it more basic or more acidic? Right. And so because the acid pH down, woo, I can't look at that. I hope you guys aren't having the same freak out moment as I am. So the pH is down, and so that's going to release, right, your acid hydrolase into your late endosome. Acid hydrolase released. Okay, right, because the pH is down. Here's the pH is higher. I wouldn't say high. It's like less than 1. It's like going from like 6.5 to 6 or something like that. So this is like a pH of 6, and this is like a pH of 6.7 or something like that. But that pH change is enough to disrupt the interaction between the acid hydrolase and the receptor. So what's this acid hydrolase going to do now? Is it going to get degraded? No. Okay, good. So eventually the pH is going to go lower, and it's going to make its way to the lysosome. pH lower. It's like 5, right? And that's where it's going to function in degradation of whatever gets sent to the lysosome. Okay? Oh, ugh. There we go. I'm going to try to stay away from it. That's horrible. Do you guys like the board better? I don't know. How about the people in the back? Is the board hard to see when I draw on it?
No, it's fine.
Who's saying it's fine? Okay, got to find from the corner. How about this corner? Do you like when I draw on the board or on the, like, make you puke Elmo? Huh? Only when you draw on this side. Okay. Well, now it kind of stopped freaking out. So I guess I just need to not go in and out of it too much. Okay, that was fun. Any questions about this? Did everyone kind of get this when they did this? So I want you guys to really be either drawing out or writing out. Some people are visual learners, so draw out processes with all the details in it. You'll remember it when you do something with it. If you're the kind of person that needs to write things a million times, make this into some kind of narrative. But I want you guys to be thinking whole process. Okay, so if I do this, go away. Oh, I know what it is. Sorry. There we go. So that was what we just did. Okay. So I put this up. Every now and then I like to find little cartoons, keep things light and fluffy. So this is a very exciting professor saying, after years of intensive research, is there something we need to talk about? Sometimes I hear discussion and it makes me stop, and I want to make sure we're all okay. Are we all okay? Everyone's okay. Okay. After years of intensive research, we finally have a clear picture of insulin action. You got your insulin, and then the receptor, and then something happens, and then there's some effect. And so this is kind of our introduction to receptor-mediated endocytosis. And I think it's funny, because the little guy saying a long way since the black box concept. And here's my other funny slide. Of receptor-mediated endocytosis. The R is the receptor, and then the little froggy is the ligand, and then you have this clustering of receptors into the clathrin-coated pits, and they're all like, this one's in love. And then, coated pit! And so the clathrin-coated pit is forming, and they realize that things aren't going well. And so what they're showing here is going to the lysosome, which is kind of like getting us ready for Halloween. Who's ready for Halloween? I'm not really. You are? Okay. I feel like it's still summer, but the stores are telling me it's time for Halloween. And I just got a catalog in the mail that was telling me it was time for Christmas, and that weirds me out. It told me I needed to buy Christmas ornaments, but I didn't want to. It was weird. Okay. Sorry, guys. It's like too late in the day. Okay, so then you've got your, what's going on here? You've got these receptors going back to the cell surface. And I want you to kind of take note of that, because that is accurate, because a lot of the time you have your receptor and ligand coming in, and then in the lysosome the pH drops, and then receptor and ligand separate. Look at that. It's so accurate. I love it. And then the receptors kind of butt off and head back up in the recycling endosome, back up to the plasma membrane. So that's exciting. It is scary, isn't it? Look at that. It's scary. Okay, so that's my introduction to endocytosis. So so we've done all the ER Golgi and outwards, and now we're going to go inwards. So endocytosis from the cell surface inward to the lysosome. Do proteins such as acid hydrolases go from the cell surface to the lysosome to work? No. Okay, just checking. So this is a cell exterior, early endosome, late endosome, lysosome. And really early endosome, late endosome, lysosome, the only difference is the pH is getting lower and lower and lower as you get to the lysosome. So this is just showing different forms of endocytosis that you may have seen before, such as phagocytosis. This is a neutrophil, but white blood cells tend to have the ability to phagocytize. And this is where they're actually bringing in full bacteria. Here's 2 bacteria are being brought in. What kind of bacteria is that? Not cocci. Okay, it's some kind of rod. I don't know. It's a trick question. Okay. Pseudopod formation. So this is a pseudopod. So you can see the cell membrane is kind of reorganizing itself to come up and over the bacteria. And I just wanted to point out that formation of the pseudopod is driven by actin polymerization and reorganization. And the reason I'm pointing that out now is because it's going to connect later on in the semester when we're talking about the cytoskeleton, and we'll talk about how actin is reorganized for cell movement and stuff like that. And this is one example. So just kind of store it away, and maybe this will come back in I don't know, like 2 months or something. Okay. So endocytosis, one form of endocytosis is through clathrin-coated vesicles, and we've already talked about clathrin. And so this picture is straight out of the book, and it's showing the uptake of lipoprotein into a hen oocyte to form a yolk, which is kind of cool. And so here's your clathrin-coated pit, and the vesicles forming. And on the outside, you can actually see the lipoprotein bound to receptor, this fuzziness over here. And so it's all on the inside. And that's what's in a yolk, right? Yeah. Cool, huh? No, sorry. I'm going to have to stand over here. Okay, so then there are also caveolae, which they form pinocytic vesicles. So this is, rather than using clathrin or COP1 or COP2, these coat proteins to form the vesicle, they invaginate based on their lipid composition, which is another way. These are used in pinocytosis, and so that's where it's just this small sampling of the extracellular fluid. And so they're membrane domains that are enriched in these GPI-linked proteins. What are GPI? Remember we talked about GPI anchors and GPI anchored proteins, and how that if you have a GPI anchored protein that is added on in the ER. Remember that a little bit? Yeah. It's also these membrane domains are rich in glycosphingolipids, cholesterol, and caveolin proteins. And so this is just EM, and it's showing the caveolae. They said in the book it looks like cauliflower. Do you guys think it looks like cauliflower? These things? A little bit. Like cabbage? Cabbage. Actually, you're right. It looks more like cabbage than cauliflower. And then here's a clathrin-coated pit, which has that basket kind of shape. So that's a nice picture. Okay, so some other fun facts about caveolae is some viruses like to enter their host cell in vesicles derived from caveolae. So here it's showing SV40 and also papillomaviruses. They both like to enter host cell through caveolae. And then they'll escape and get to wherever they need to go and make trouble. A lot of viruses get in through receptors, so sometimes through receptor-mediated endocytosis, and then this is just through caveolae. Yes?
What triggers them to, I guess, create
What's the mechanism? I don't know the mechanism off the top of my head, but it would be very easy to look up. Shall we make that a stop the teacher question? Okay. What is the mechanism for SV40 or papillomaviruses to get into the cell, right? Because we know it's through caveolae, but how? What do they do, right? How? So if you tell me it's through caveolae, you will not get points because I told you that. So I want a mechanism. What is it binding to? What is it? How is it inducing endocytosis through caveolae? Okay, and that is due tomorrow by 4 p.m. Okay. So this example here is just showing that viruses get into the cell. Some viruses, SV40 specifically, and papillomavirus get into the cell through caveolae. And so the question from Tyler was how? What's the mechanism? And I can come up with some ideas, but I don't really know. So look it up and tell me what you find. It's possible that looking at this paper might tell you something, because there's a reference right there. But I would also suggest that you look a little more deeply, because I'm not really sure. Okay? Mechanism. Good question. Okay. Where have we been and where are we going? So we're gonna like zoom back out for big picture, a little big picture break, right? So we were talking about proteins coming out, RNA coming out of the nucleus and associating with the ribosome, and then it needs to go somewhere. And so right now we're on the secretory pathway, where we have ribosomes making mRNA, and then it gets into the ER, the Golgi, and then either the plasma membrane or lysosome, and that's the secretory pathway. Next week we'll be doing the rest of this, which will be to the mitochondria. I kind of ignore the chloroplast, I'm sorry, plant lovers, and nucleus. I mostly stay with the nucleus and mitochondria. So that's where we're going. But let's stop for a minute and finish up the endocytic pathway with receptor-mediated endocytosis. And so all of you guys read about that this weekend. So I really don't need to teach you anything, right? So we can go? So what is receptor-mediated endocytosis? Is it phagocytosis? No. Yes?
Selective binding for internalization of particles.
Selective binding and internalization of particles. That's a good one. Does anyone want to add to that?
It's a mechanism for selective import of macromolecules into the cell.
It's a mechanism for selective import of macromolecules into the cell. How is it selective? What makes it selective? Okay, right. There's a receptor. So those receptors are specific. And there's specific binding between a receptor and a ligand. So this is the basic picture of receptor-mediated endocytosis. This is out of Molecular Cell Biology, which is Lodish's book. It's also on NCBI books, so feel free to pull this up, read the blurb that goes with it. Because they had a really nice picture. This is just kind of the life cycle of a receptor, using LDL as an example, where you have your LDL receptor up top, and then your LDL particle will bind to it, and then there'll be formation of the clathrin-coated pit and endocytosis, right? And then your clathrin will go away, and then you've got your vesicle, which becomes your early endosome. And then you have your late endosome. So actually here it's showing your early and late endosome fusing. And then you have this, what you have happening here is this recycling endosome forming. And so as the pH starts to go down, your LDL will be released. Oh look, there's a pH thing again. Your LDL will be released from your receptor. And then those receptors will kind of all congregate in the same area that will form a recycling endosome. And notice there's nothing specific actually here to coat proteins and stuff like that. But I would assume that there is something involved. And so you have the recycling endosome that goes right back up to the plasma membrane. And then that leaves your LDL particle to go on and fuse with the lysosome. And then the lysosome, the pH is so low that the LDL can then be digested into all of its parts, which is phospholipids and protein and cholesterol. And that's what needs to stay in the cell and be used, right? So why is it important for there to be receptor-mediated endocytosis versus pinocytosis or some other non-receptor-mediated endocytosis? Yes?
Maybe you're running something in particular and express those receptors and then bring those in versus other metabolites and necessarily.
Right, so there's a level of specificity. You can express certain receptors that are going to bind to certain ligands to bring in what you need. Right. Whereas pinocytosis is just kind of this non-specific endocytosis, and phagocytosis is particular for specific for bacteria and stuff like that. Right, so it's specific for whatever the receptor will bind to. So what if there were a mutation in the gene encoding clathrin? What would be the effect on receptor-mediated endocytosis? If it was a mutation that prevented clathrin from interacting. Yes?
Would it not form a little vesicle thing?
Right, so it would be able to form vesicles. Did I see another hand up over here? Right, because clathrin is about formation of it's the actual pit formation, the actual vesicle formation, and clathrin is interacting with the receptors. And then if you can't get interaction between clathrin and the receptors, well, through adaptin, then you would not be able to form the vesicle. Yes? Pinocytosis or endocytosis? Pinocytosis. So the question was, what is pinocytosis? Is it random? I think it is kind of regular endocytosis of the liquid material surrounding the cell, kind of to help to sample the environment. It's not specific. So what if there were What if there were a mutation in the genes encoding the proton pump in the late endosome? What would happen? So the late endosome is pretty much over here. So if the proton pump had a defect in it, yes?
It would change the pH of the vesicle.
It would change the pH. Okay, and how would that affect what was in there? What if you're pumping protons in, yeah?
Hydrolases wouldn't function.
Hydrolases wouldn't function. So that's in the lysosome, right? So hydrolases would not function in the lysosome. Yeah.
The receptor wouldn't dissociate from the ligand.
The receptor wouldn't dissociate from the ligand. Is that what I was hearing coming out over here? Yeah. Yeah, because it's that pH drop that causes the receptor and the ligand to dissociate. Right. And so in the lysosome, just like what you said, these acid hydrolases would not be active. And so what would be the effect on LDL? It wouldn't break down, right? It wouldn't break down because it's that drop in the pH that activates all the acid hydrolases that then degrades LDL into all of its parts that are used by the cell. Okay. Yes.
So wait, you're saying the mutation wouldn't lower the pH, which and then would...
Right. So the proton pump. What does a proton pump do? pumps hydrogen ions into a cell, and that would decrease the pH,
Okay.
right? So it would become more acidic. That's the mechanism for acidification. Yeah. Okay. Any questions about this? Okay. So this is just what it looks like close up. You've got your LDL receptor, and the LDL receptor is going to interact with the adaptin in the clathrin, just like what we've learned. So what would happen if the LDL receptor had a mutation that resulted in a premature stop codon in the cytoplasmic domain? So I'm going to give you guys a minute to talk with a friend about this, because not everyone can just be like bam, bam, bam answers. So talk about it with a friend and tell me what would happen. What would be the result of, in terms of receptor-mediated endocytosis and the fate of LDL and all that? Talk for a minute.
So essentially, basically what that's
Yeah, that's right.
So what it means is, so I just had a question about what do I mean by a stop codon in the cytoplasmic domain. So this is your extracellular domain, and here's your cytoplasmic domain. It's your cytoplasmic tail. And so that there would be a premature stop codon, so this bottom part wouldn't be there.
Oh.
Yeah.
I didn't think the cytoplasmic part is fine. So it's just kind of like a receptor that can't move because of no function of cytoplasm. Or can't function.
Okay, guys. It seemed like that clarification really helped. So, what would happen if the LDL receptor had a mutation that results in a premature stop codon in the cytoplasmic domain? Yes?
The clathrin wouldn't be able to bind to it.
Right.
So it wouldn't be able to form a vesicle to bring the LDL.
Right. Clathrin wouldn't be able to bind because that's where adaptin is binding. right? And so it couldn't form a vesicle, couldn't bring LDL into the cell. That's not good, right? Yeah. Yep, so it brings in, yeah, low density lipoproteins. Yeah. So it's like your source of getting cholesterol and phospholipids. You want to bring it in so you can use it to do things like make membranes. Yeah. Yep.
How can we assume that a premature stop codon would only affect the cytosolic side and not the entire?
So the question is, how can we assume that the premature stop codon will only affect the cytoplasmic domain? Well, it depends on where it is. So I'm telling you in this question that it's in the cytoplasmic domain, so it should stop translation before you can make your cytoplasmic domain. But it all depends on the location of your premature stop codon. If it was after the first 5 amino acids, then you'd never have anything. Right? Yep, good question. Any other questions? Okay. Okay, so what are there any clinical implications of a defective LDL receptor? What do you think might happen when you go to the doctor and they take your blood if you have a defective LDL receptor? You'll have a lot of fat in your blood. What else will be high in your blood?
Sorry.
Cholesterol, right? Right? Because you're checking your blood for cholesterol. You'll have cholesterol in your blood is not a good sign. Right? They tell you that's bad. Yeah, so that's fine. Okay. So here's another virus example of endocytosis. So HIV, I love viruses, can you tell? It's so funny because I hated them for so long. And then I ended up doing my PhD in a virology lab. It's weird. So just goes to show you what you think you love now, who knows what's going to happen. So HIV NEF downregulates CD4. Does anyone know what CD4 is?
It's a receptor on a helper cell.
It's a receptor on a helper T cell. Do you guys know what T cells are? I don't mean to talk at you, but not everyone loves immunology. It's the cells, they're white blood cells that help to fight infection, but CD4 positive T cells are the target of HIV virus. But they also, CD4 happens to be involved in activating the B cell side, and the antibody response. So HIV virus downregulates CD4 by inducing endocytosis of the HIV receptor. And actually what it does is it has its own protein that kind of acts as an adapter protein and interacts with clathrin and CD4 and basically induces endocytosis that way. And so that's kind of cool if suddenly the main mechanism for one of the main mechanisms for activating part of the immune system is shut down. That's pretty cool. Well, not really for us. It's cool for the virus, right? And so then it goes to early endosome and it's degraded in the lysosome. Okay, so who cares about all this? You all should care about all of this now because I've told you about how exciting it is. Signal sequence. Oh my God, another signal sequence. So there's a signal sequence that determines the recruitment of a plasma membrane protein. So it is how plasma membrane proteins are recruited into clathrin-coated pits for endocytosis. And it is involved with the interaction between the membrane protein or receptor and adaptin. So the endocytosis, it's called an endocytosis signal, and it's YXX hydrophobic. It's actually not very It's a consensus sequence. It's not very specific. It's not like HADL. HADL is so obvious, right? But this is tyrosine, any amino acid, or any polar amino acid, and then any hydrophobic amino acid. And then the LDL receptor is an exception that has a unique signal sequence. Don't worry about memorizing these things, but do understand that there is a sequence in the cytoplasmic tail of receptors that is involved with interaction, specific interaction with the adaptin and clathrin. Okay? So there are many different receptors that can accumulate in a clathrin-coated pit. So it's not just LDL, or just, so these clathrin-coated pits can actually bind to a lot of different receptors. Okay? And so why would that be? So protein-protein interactions are really important for clustering of receptors into a clathrin-coated pit, right? Now I'm telling you that a lot of different types of receptors can be incorporated into a clathrin-coated pit. Why would this be? What mechanism is responsible for that? What protein-protein interaction? What's conserved? What's similar? Oh God, a lot of really confused looks. No, I'm not repeating anything. No. So I'm telling you, so right here you have your LDL receptor, and then you have transferrin receptor, then you have some other receptor. They can all be in the same clathrin-coated pit. Why? How? What interaction is responsible for allowing many different receptors to accumulate in a clathrin-coated pit? Yes.
They all interact with adaptin.
They all interact with adaptin, right? Through what? Yes.
You have the same sequence?
Right. They all have the same endocytosis signal. So there's a conserved interaction there, right? Adaptin binds to this endocytosis signal. As long as they have something that will bind to that region of adaptin, they can all be brought into the same clathrin-coated pit. Any questions about that? Okay.
I'm sorry, did you say that they all have the same signal sequence? Is that what you just said?
Yep, so there's this endocytosis signal.
The endocytes.
Yeah, that is on the cytoplasmic tail of the receptor, and that's interacting with adaptin. And that's why a whole bunch of different receptors can all accumulate in the clathrin-coated pit. Okay? Yeah. Can you assume they'll all be localized in the same spot? Expand on your question.
Everything else that's responding to those receptors are all below the same area.
Okay, so you're thinking, how do they all get to the same spot on the membrane so they can be endocytosis? Is that what you're thinking about?
They're all going to be in the same vesicles, and they're all getting shipped in the same spot.
Well, they're all going, where are all these vesicles going?
So it's just lysosome.
Eventually, right? So if you're supposed to be recycled, you'll end up in a recycling endosome, which will go back up the membrane. Otherwise, you're going to the lysosome, so it doesn't really matter, right? Okay. Good question. So you might be saying to me, but wait, how can you regulate what gets endocytosed and what doesn't? You're saying all receptors can get recruited into clathrin-coated pits. Well, how is this? You should be asking me, how is this regulated, right? Everything's regulated. It's not just like, oops, endocytosis. Well, maybe. Some things actually, I should take that back. Some things do kind of have a regular schedule, but a lot of this is also regulated through monoubiquitination of receptor cytoplasmic tails. So you can have a receptor and it's being recruited into the clathrin-coated pit, but that's because you have ubiquitination. So the cytoplasmic tail is ubiquitinated, but monoubiquitinated, not lysine 48 polyubiquitinated, which goes to the proteasome, not that one. Remember I told you there's all these different kinds of ubiquitin that do all sorts of things, and there's one type of ubiquitin that when one molecule is added to a cytoplasmic tail of a receptor, it signals for recruitment of the endocytic machinery. And then recruitment of adaptin and clathrin and all that stuff. And so that helps with regulation of endocytosis.
Wait, do you mean that when it's ubiquitinated, it's recruiting?
When it's ubiquitinated, monoubiquitinated, it recruits that endocytic machinery and then is recruited into a clathrin-coated pit and endocytosis. Yeah. And this is not ubiquitination that sends something to the proteasome. It's a different kind of ubiquitination. So that's why I keep saying monoubiquitination, not polyubiquitination. Okay? Okay, so we're going to have a practice problem, so we can all take a breather. So you're studying a new receptor. Are you ready to study a new receptor? Nope. Okay. You are trying to determine whether it participates in receptor-mediated endocytosis through clathrin-coated protein. You know the protein's amino acid sequence. How can you use recombinant DNA technology, meaning cloning something, cleaving something, cutting something, pasting something, to address these research questions? So the question is, what's the question? The question is, does this receptor participate in receptor-mediated endocytosis through clathrin-coated pits? That's your question. Sometimes you just have to search in there for the question, right? Okay, so does anyone need clarification before I give you a minute? So you need to ask yourself, what's required in order for a receptor to participate in receptor-mediated endocytosis through clathrin-coated pits? There are a few pieces of information I just gave to you. Okay? Just to be nice, I talked to you about an endocytosis signal. I talked to you about ubiquitination. I talked to you about interaction with adaptin and clathrin. And so there's all that information I want you to use to try to solve this, come up with a method to determine whether or not your protein participates in receptor-mediated endocytosis through clathrin-coated pits. Okay?
So you know the sequence. Okay, so this side is always side. In terms of so basically, you know the sequence. You know everything it does participate in it, but not even. And then you're going to refer to that as
Right, you were being received.
But you do. You have you know it's a sudden solid. So if you have so this is comparing a lot of different. Let's say you have the same sequence, you put it on another receptor which you know doesn't participate in and then you pick from that receptor and see so basically what you are saying is that okay, this specific so this is one sequence and the sequence is, you know, part of the data that needs to come from another receptor. If
I can't think of another way. That's the right. Well, yeah, yeah, it's on the side.
That's not the, I wouldn't find calculators in what the trade muscles I'm saying
What do you call
I'll learn the instructions.
Okay, guys, are we ready or do you want another minute? I see one thumbs up. You can never tell how long I should give you. Am I giving you a good amount of time? Okay. Sometimes I see like 5 people discussing. I don't want to stop it. Okay. Okay, so tell me, what did you guys come up with? I know people came up with something. No one ever wants to look dumb. That's the problem, right? Right? But it's okay because they'll just say, no, you're wrong. And I'll say it with a smile and then we'll move on. Yeah.
I said if there's an F in the signal sequence that we talked about, you could include that and see if just below somebody's
Okay, so let me go back here. So this endocytosis signal. You're saying, can you remove that and see if you still get endocytosis? Okay, that's one. That's a good one. Yeah.
Could you also kind of do the inverse of complete it and put it on something else and see if that still.
Can you add endocytosis signal to something else and see if it's still formed? Right. So that will tell you. So what you're really doing there is determining whether or not. Well, first of all, what do you have to do? Do you know if there's an endocytosis signal? So I told you, oops, that's the wrong way. I told you that you know the protein's amino acid sequence. So the first thing you have to do is look to see if there's an endocytosis signal. And then you can use your recombinant DNA technology to engineer a protein lacking sequence and see if it goes where it goes. Or take that sequence and stick it onto something else that shouldn't, maybe that goes into some other mechanism and see if you can force it in that way. Yeah.
Or if it's like LDL, it has a unique signal sequence.
Right, right, and that could be the problem, right? So what if it's like LDL and has a unique signal sequence? What would you do? Because I'm sure the people who are studying that first looked for the consensus sequence, and then we're like, oh my God, what do we do? So what would you do then? you'd have to get much more novel, right? Right? Yes?
If you cleaved off the sequence that makes it receptor mediated and still forms a vesicle, does that mean it's doing some other type of endocytosis?
That could mean many things. That could mean it's doing another kind of endocytosis. So the question was, what if you cleave off this endocytosis sequence and you still get receptor-mediated endocytosis? Does that mean it's going through some clathrin-independent mechanism? It could mean multiple things. It could mean that, or it could mean it has some kind of novel sequence like LDL. That could have been how they discovered it. One of those two, you'd have to do more experiments. So let's go back to, whoa, somebody, okay, yes.
No, I just have a question. When you're talking about cleaving something, what are you specifically talking about cleaving and putting somewhere else?
So you can actually take, so the question was, when you talk about cleaving something, what am I talking about?
Yeah.
So when you do these kind of experiments, you usually are cloning a gene into a vector. And so you can actually engineer whatever you want. And so when you're doing your cloning, you PCR up whatever part of the protein you want,
Right.
and you paste it into your vector. And so in this case, your sequence of interest, let's say it's this C-terminal endocytosis signal sequence, because you don't know where it is, but it's in the cytoplasmic domain. So then what you can do is you can engineer a protein that's lacking that bit just by creating your PCR primers, excluding that, moving a bit downwards. And then that, or let's say you want to see if some other protein, you make some fake protein and see if you can force it into clathrin-coated bits. Then you can actually, through PCR, add on your endocytosis signal to some other protein.
Right, no, I get that, but that's a part of the protein, right? So then you would then run the protein like through this receptor, the new receptor that we're trying to figure out if it's a part of the receptor-mediated endocytosis. So you're talking about cleaving part of the protein?
No, so remember you have protein, you have DNA, RNA, protein.
Right.
So all the engineering gets done at the DNA level,
Okay.
and then you put the DNA into a cell, and the cell makes RNA and makes protein, and the protein it makes is whatever DNA sequence you put in.
Okay.
Does that make sense?
A little bit.
We can talk more about it.
Okay.
In the back. Yeah. You, then you, because I saw you first, and then you.
So, even if you cleaved off the sequence, would that necessarily tell you the like part of receptor-mediated endocytosis? Is there a bunch of different receptors all lumped into one? So you can still form endocytosis?
Yes, you'd have to be, yes, so the question is, even if you removed it, would it tell you if it's involved in receptor-mediated endocytosis? And so you would have to find a way to follow your receptor, right? So the ideal thing would probably be making it a GFP tagged receptor, and then you can look through microscopy and see, so you put it in the cell, and you say normal protein seems to be internalized off the surface of the membrane, and the protein where your sequence was removed is no longer internalized, it stays on the membrane.
So you'd be able to see that receptor down to something? I mean, because
Well, this is about a 10 experiment thing that you have to do to really prove your point, right? So one would be, but you'd start by making these mutants, and then you have to carry out all these experiments. One would be, is it getting into the cell? Another would be, is it interacting with adaptin? Is it interacting with clathrin? And that would all be immunoprecipitations, running gels, doing Western blots. And so you can't usually do one experiment to prove your point. Yeah, it would be like actually a whole paper of experiment. About 3 years of work to prove that. Yes?
Is that all the receptors have the same common endocytosis signal, and they all bind to the actin, right? So LDL has
Yeah.
so adaptin still binds to them.
Yeah, but still, so there's always exceptions to the rule. So I put that in there because it just so happens LDL has some non-canonical sequence that also participates in clathrin-mediated endocytosis through adapter. Yeah. Yep. And so probably what happened is they started out by cutting off the consensus sequence, and then they saw it was still internalized, and they were like, what's going on here? And then through a million experiments, decided to start doing mutations all over the place in the cytoplasmic tail, and then found that it was still getting in until you hit this one sequence, and that's what was responsible. Yep. Any other questions? Okay. Is that a fun exercise? A little bit? Okay. Okay, so unless cargo is diverted elsewhere, the default pathway for molecules taken through endocytosis is the lysosome. Okay, so you can get diversion by recycling back to the plasma membrane. So that's the recycling endosomes. And then you can get also transcytosis, which is transit of receptors to a different plasma membrane of the same cell, and that's really in polarized cells which have apical domains and basolateral domains. And then you get degradation in the lysosome. So those are the different fates of receptors that are brought in through endocytosis. So the LDL receptor is an example of recycling, where you'll have endocytosis through clathrin-coated pits, fusion with the endosome, and then you've got in the early endosome these budding of these recycling endosomes that will go up to the plasma membrane, and then the rest goes to the lysosome. The LDL receptor is a little bit different in the sense that it's not so much that something binds to the ligand and then it's internalized. It's automatically internalized on a regular basis. Don't ask me the mechanism. Every 10 minutes or so, regardless of LDL binding. And I guess we can kind of come up with a guess mechanism if we want to. My guess is it probably, well, I won't go there. And so then you have to ask yourself, what are the fates of the LDL receptor containing endosomes, right? So you've got these endosomes and the receptors going back up to the plasma membrane. Sometimes I put these questions up here so that when you're studying, you can remember what to ask yourself. Any questions about this? Okay, so here's another example of receptor-mediated endocytosis. And this is different. So I want you to, in your mind, compare and contrast the difference between the LDL receptor and the transferrin receptor. So this is an example of recycling the receptor and the ligand. Okay? And that's because transferrin binds iron. And so there's no need to get rid of the transferrin over time. So you can actually bring in your receptor plus your transferrin, either in the ferrotransferrin, so that's iron-bound, or apotransferrin, which is no iron-bound. And then the iron's released upon acidification in the late endosome, and then the transferrin receptor plus apotransferrin is recycled back to the plasma membrane. Right, so here you're seeing ferrotransferrin. This is this iron-bound transferrin bound to the receptor, and then you get endocytosis, and clathrin-coated vesicle becomes an endosome, then a late endosome, and as the pH goes down, you get dissociation of the iron from the transferrin, but the transferrin is still staying bound to the receptor. And then the apotransferrin, now free of iron, goes back up and with the receptor, and then apotransferrin is dissociating at a pH of 7 and goes off and finds more iron to bring back. So here there's two themes here. One is just a general theme of receptor-mediated endocytosis and how there are different ways, and then we're still with the pH theme. And here it's even different, right? Here you have binding of ferrotransferrin to the receptor, which then comes in, and then at a low pH, only the iron dissociates. And then at the pH, as the pH increases, you get dissociation. So it's different. And this is because every protein-protein interaction is specific and has its own optimal pH. Okay? Any questions about this? Are you guys done? We're done today. Okay, so let's just finish this slide and then I'll let you leave. So why is this an economical way for the cell to take up iron? Right, you always want to think about economy when you're thinking of science. Does the cell want to use excess energy? No. Right, so by recycling not only your receptor but also your transferrin, you're using less energy because you're not having to constantly do transcription, translation, all the stuff that you need to do to make your transferrin over and over and over again. Okay? How does this differ from the LDL receptor? Is this the same or different from the LDL receptor? Different, right. Why? Come on, give me one more before I let you go. Here, let's go back. Because you're getting recycling of your transferrin, right? Great. Okay. Go away.